Yeah.
Hello, we're group 11 and we're gonna present about genetic inheritance. My name is Eros Barahona.
Dhruv Champaneri.
Aidan, Dyer.
Khansa.
And I'm Aditya Tigadi.
So what is epigenetics? For a quick overview of epigenetics, it is the study of how environmental and behavioral factors impact gene expression without changing the genetic sequence. So what determines our epigenetic is our epigenome. The epigenome is a set of chemicals that surround the DNA. These chemicals determine the different types of regulation that will be taking place. So for the type of regulation, one common type of regulation is through non-coding RNA. These RNAs do not translate to proteins. Instead, when they see an mRNA, they attach themselves to the mRNA. That way, the gene is not being expressed, therefore silencing the gene.
Now, there are three types of regulation pertaining to DNA. The first is methylation, which inhibits gene expression. By making the DNA wrap around histone proteins more tightly using methyl tags or methyl groups, the DNA becomes unreadable, silencing the gene. Acetylation utilizes acetyl tags or acetyl groups to loosen the DNA around histone proteins, giving polymerase enzymes space to attach to the DNA. Thus, the expression of the genes in that area are boosted. Phosphorylation contributes to condensation of chromatin during mitosis, influencing DNA's ability to proceed in reactions. It is a response to DNA damage. It delays the progression of the cell cycle, thereby giving more time to repair the damage. Now, how are these modifications obtained? Almost anything we do affects our epigenome. Stress factors from work or personal life can release hormones such as cortisol that can initiate methylation or acetylation. Diet and nutrition during pregnancy plays a major role as well, considering the fact that all of the building blocks of our cells, including our DNA, comes from the food we consume. Chemicals from pollution or smoking can enter our cells through the respiratory and circulation systems and can cause reactions to take place that directly affect the epigenome. Additionally, age and disease are just as important. As we grow older, changes in our hormones of genes at specific loci. Depending on our environment, we may encounter different bacteria or germs that can influence our genetics through various ways, such as altering the structure of chromatin.
As we saw how different environmental factors and behavioral factors affect our DNA, now we will dive deeper into the specific examples based on real-life studies. According to a study in India on arsenic level exposure, it showed that higher arsenic levels cause skin lesions. As you can see in the middle image, you can see this individual that has skin lesions. Scientists saw that arsenic induced hypomethylation, which led to carcinogenesis or formation of cancer cells. This occurred primarily on the repeating DNA sequence, also in some other regions, other gene regions. Hypomethylation has less methyl groups attached to the genome, and whereas hypermethylation has more methyl groups added to the part of the genome. As you can see on the left image, the normal cells are converted into tumor cells. So in the normal cells, there is regular methylation, but in tumor cells, there is a hypomethylation on the CPG sites or DNA sequence that has primarily made up of cytosine and guanine. So this hypomethylation leads to tumor cell formation. Hypomethylation also in general arises earlier and is linked to chromosomal instability and loss of imprinting. White circles, as I mentioned, were the unmethylated CPG sites, and red circles show methylated CPG sites. Hypermethylation also leads to gene silencing because you have more methyl groups attached, as you can see in these red circles. So it promotes less transcription or gene silencing. As we all know that air pollution is dangerous for our health, it contains particulate matters with varying sizes and other different particles that are harmful to our health. As you can see in the diagram on the far right side, it's a size comparison of particulate matter of different microns or micrometers with compared with human hair and a grain of sand. In a specific study on particulate matter of 10 micron exposure, it was seen that it promoted demethylation on a gene that was a key player in inflammation and oxidative stress, which overall led to cardiorespiratory diseases. Airborne benzene, or as it is known as aromatic solvent or organic solvent, was found to induce hypomethylation on a cancer antigen gene that would produce more cancer cells. From early studies, it was very clear that DNA methylation was sensitive to environmental stress exposure in early developmental life. Glucocorticoid gene receptor was studied in different stress responses. So why talk about glucocorticoid receptor? Well, if you remember from your general biology class, you would remember that glucocorticoids are a class of steroid hormones that, when bound to its receptor gene, cortisol hormones are released in stress response. Fat breakdown, amino acids, and glucose are released in the bloodstream because our body requires energy source, and that comes from glucose. Hypermethylation. There are different types of there are two different distinctions in these receptor gene: hypermethylation and hypomethylation. Hypermethylation occurs in stressful environment. According to studies, hypermethylation was found in higher amounts among suicide victims with history of child abuse, but not with suicide victims with no history of child abuse. Hypomethylation also showed that in animal studies, high maternal care induced hypomethylation of glucocorticoid receptor gene in the hippocampus of our brain. The image that you're looking at on the left side is a molecular structure of glucocorticoid, and on the right side, it's showing how positive parenting is very necessary for an infant or a baby at a very early age, because positive environment promotes a proper development of a baby.
Building on what Dhruv was talking about in the last slide and the role epigenetics plays on psychology and mental illness, some studies were done, some epigenome studies had discovered several sites with DNA methylation altercations in genes which are involved in brain development and neurotransmitter pathways. So this can be associated with mental illness that started at a young age and can cause lifelong disability. Epigenetic events which alter chromatin structure and change the gene expression have also been associated with depression-related behaviors, depending on which gene has, been affected. In a study done on mice, social avoidance resulted in decreased expression of hippocampal genes, which is important when mediating depressive responses. And there was another study done that observed the brains of individuals with a history of clinical depression. So they took post-mortem brain samples and they found level of histone markers of increased gene expression, which were down-regulated in those brains. And those findings were consistent with the previous findings done on the study with mice. Next slide, please. Also, epigenetics plays a role in learning and memory. Epigenetic mechanisms have been shown to influence genomic activities in the brain to produce long-term altercations in synaptic signaling, organization, morphology, all of which are structures which support memory and learning. Changes in DNA methylation and disruptions to gene encoding in the DNA methylation machinery can cause learning and memory impairment. Changes in histone modifications have been shown to influence long-term memory formation by altering, again, the chromatin structure and the expression of genes corresponding to learning and memory. So one example of this is seen in Rett syndrome patients, where there's a mutation in the DNA sequence called MECP2. So MECP2 plays many different roles, but particularly reads DNA sequences, checks for DNA methylation, and then binds to those areas which contain those methylation and allows certain proteins to, regulates the protein. So in a study done with mice with altered MECP2 expression, scientists found that there were increase in histone acetylation, neuron cell death, increased level of anxiety and cognitive defects, all of which are common and support disruptions that may be found with Rett syndrome patients.
So we can look at the effects of epigenetics in real life. In these experiments, we can see 2-3-year-old twins. And in the first picture, we can see that they have the same genetic code as well as the same epigenome. They have the same chemical tags around them. However, after a life, we can go back to them when they're 50-year-old twins, and we can see that they have different epigenome. They have different chemical tags surrounding their DNA because they have been exposed to different environmental factors and life decisions. For our thought experiments, we can dive into this. We can say that we have a couple of clones babies, but they are nurtured differently. They have different diets and activities, as well as taking different life decisions. And more importantly, they live in different environments. By the end of their life, we're gonna end up with different individuals that have the same age and they have the same genotype. However, they will have different hobbies, they will have different health conditions, they will have different phenotypes. That was, they all look different. This is because even though they have the same genes, those genes were affected by their environment differently.
Now, one might ask, are these epigenetic changes heritable, or are they passed down to future generations, and are they reversible? Well, first of all, we need to know there's a specific phenomena behind this. It's called transgenerational epigenetic inheritance. It is a transmission of epigenetic markers from an organism to another, or from a parent to the offspring. The environmental factors affect the epigenome of an individual in the somatic cells, but not added over our genes. Usually they are cleared, but sometimes, usually the epigenetic markers are cleared, but sometimes they are left at some specific loci in the genome that can be passed down to the future generation that would have the passed down epigenetic markers. Studies are still ongoing in this particular field of research. These epigenetic changes are reversible, as the genome sequence is not edited, but it's modified by different modes of regulation. As you can see in this diagram, the histone modifications are changed. It can be methylated or acetylated. It all depends on the environment, so it can be reversed. Next.
So as we said, epigenetic characteristics can be passed down through the generations. One study was able to predict cancer mortality and general mortality of the grandchildren of people that went through starvation conditions. These modifications, grandfathers that went through starvation conditions, they found had certain groups of genes methylated. The male offspring of those grandparents and those grandparents' grandchildren also had those methyl groups modified. Interestingly, these methyl group modifications that happen once starvation conditions are exposed to someone only are passed down through the male line. Right now, it's not super well understood why that is. that is an image of a methyl group modifying DNA after people go through starvation conditions. So this shows that the way that people live now can affect the health outcomes of people in the next generations, even if it's only passed down through the male line. In this case, in most cases, epigenetics are passed down through both male and female lines. So this just shows that health outcomes can, to an extent, not completely, be predicted and affected by epigenetics, which are passed down.
Thank you for the time.
Thank you.